is a review of Unit 2, chapters 5 to 11. And this is just a review to point out to the most important parts of these chapters. We'll start with chapter 5. And I'm not going to spend much time. I'll just point out to the most important parts. So subcutaneous layer is also called the hypodermis, and this is the location of fat and injections. Okay. Functions of the skin, you need to remember those. Epidermis is avascular, stratified, so how you get the oxygen. This is something important to know. How the dermis and epidermis fit together, and what the epidermal ridges do, which is the fingerprints. Keratinocytes, what's the function? What's the difference between thin and thick? This one layer that's making the difference between those two. This is 4 and this is 5. Which one is the different? And the location is also important. This arrangement is important. You need to remember it from the bottom up or from up to the bottom. And which layer is the difference between thick and thin? Lucidum, okay. Function of each one of those, specifically the cells. So the cells here are very important. So stratum germinativum that contain the stem, basal cells, epidermal ridges making the fingerprints. Okay. And what's the function of different cells? Okay. And I think I mentioned here, you should remember the degree of darkness of the skin does not depend on the number. It depends on the amount of melanin. Okay. So spinosum, if you see a cell, it's important. What's a dendritic cell, for example? Langerhans cells. So each strata has a function that you need to remember beside the arrangement. Corneum, for example, is a horny layer that's water resistant, replaced every 2 weeks. So that's what we have to remember here. You have to remember the difference between sensible and sensible perspiration and examples. Okay. Hydration versus dehydration is important. What happens if you stay in fresh water versus salty water? Hydration versus dehydration. What are the pigments that make your skin? It's a mix of these. So function of each layer, function of each cell. Okay. These abnormal coloration or the discoloration is important. What's cyanosis? What's jaundice? What's vitiligo versus albino? It's different, obviously. So you need to know the difference between those two. Where do we make the vitamin D and where do we activate it and what's called? Rickets versus what? Ostimulation. Rickets versus ostimulation. You have to remember that. What's the function of EGF? That's important too. Dermatitis, inflammation, that's also good to remember. What are the stretch marks? You have to remember this. What does it mean? Stretch marks. What's a line of cleavage? And I gave you a question of that before, if you still remember. When the cut is parallel versus across. Which one is better? The parallel, yes. What's contusion? This is also important. About the subcutaneous tissue, I mentioned the most important thing. It's called the hypodermis, and this is a location of 2 things. Injections and fat. Okay. Where we don't have the hair, this is also important. Erector pili muscle, sebaceous gland. This is also important from all aspects. What type of gland? What does it produce? Any gland you see, you have to know the type of secretion and the function. Any gland. Sebaceous gland, serous gland, ceruminous gland, any gland. Glands are important. For the hair, you can just know these layers. It's a medulla inside, cortex, and cuticle outside. Hair is made of keratin or keratinized. Okay, it's important too. What's the club hair and the new hair growth cycle? This is also important. Versus the two types, I mean the vellus and the terminal hair. This is important to differentiate between those two. What's the vellus hair and what's the terminal hair? What determines the hair color? It's melanocyte. Where? At the hair papilla. At the papilla, determined by genes. Sebaceous glands. It's holocrine gland. What does holocrine gland means and what does it produce? Sweat glands, we have two types. What's the difference between those? This is all important. Anything about the gland is important. What type of gland? What does it secrete? The difference between apocrine and merocrine glands. What type of secretion? What's the importance of each one of those? Location of apocrine, and we mentioned that. When does it maturate? And it's obviously different than the regular sweat glands, which is the merocrine glands. The merocrine gland or eccrine gland, these are the regular sweat glands. What's the function? And what does it excrete? Ceruminous gland. Glands are important. For the nail, you need to know these terms. What's hyponachium, what's epinechium, for example. And the process of repair is just a simple steps. Scape, granulation, which is this. Bleeding, scape, macrophage and fibroblast, granulation tissue until you repair. What's keloid? Okay, and the rule of ninth is also important. Rule of ninth. We mentioned that before. 9. The ninth. We mentioned that. It's this picture. Aging, everything deteriorates. Just take it as is. Everything deteriorates. That's it for this one. Chapter 6. What's the function of the skeletal system? This is something that you need to remember. Examples of each type of bone, flat bone, long bone, and so on. Irregular sesamoid. What's a sesamoid bone? Example. Patella. And what does it mean? So each one of these type and an example of those. What does it mean? Sesamoid is bone growth within a tendon. Okay, this is not very, it's not the most important thing, but you need to know the different parts, what's the diaphysis, metaphysis, and epiphysis. What's the basic structure for the flat versus the long bone? Which is compact and which is sponge, and what's the basic unit of each one of those, like diploid versus osteum. Osseous tissue. This is important. It's connective tissue and calcium. Okay. Each cell is very important, and we discussed that in details. Osteoprogenitor versus osteoblast versus osteocyte versus osteoclast, right? The two layers of the periosteum and the two layers of the endosteum, which is out and which is in, which one is cellular and which one is fibrous, okay? What is the calcium salts? What type of calcium salts will make the most, which is this slide here is important. Again, the cells are very important, but the basics, you need to remember the basics. Which one make the osseous tissue, which one maintain, which one is the stem cell, which one that break down or lyse the matrix. And what osteoid means, which is before calcification, osteoid. So each one of these cells is important, and the location is also important. How exactly does the osteoclast break down the bone? It breaks down two things, as you see, acid and enzymes. The acid will dissolve the matrix, which is the salts, and the enzyme will dissolve the protein. Okay? Osteon, obviously, the basic unit, I mentioned that. What's the basic unit for compact bone versus spongy bone? What's the difference between red and yellow bone marrow? Again, I mentioned the difference between the structure of periosteum and endosteum, the two layers. What in each layer, specifically the inner one, which is cellular, and what does it do? What are the types, specifically in the inner layer? What type of cell in the inner layer? Endosteum and periosteum. What's the difference between osteogenesis and ossification? This is different. Osteogenesis, bone formation, ossification, and when you replace tissue to another type of tissue. Calcification versus ossification. And endocardial ossification versus membranous ossification. It's important to remember what type of cartilage here, and just the main steps. No much details needed, like how does it start and grow. So just the main steps. That's oppositional growth, which is become thicker and stronger, basically. What's the difference between epiphyseal cartilage and epiphyseal line? Important. Dermal or membranous, just the main steps. Come from a membrane and give me examples. This is the most important thing here. Endocardial, there are two ways of ossification, right? Endochondrial, which start as cartilage, membranous or dermal, which start as a derm or a membrane. You need to give me examples, like dermal, mandible, and clavicle. The endochondrial is basically most of the long bones, basically. Details is not the most important thing, but you just need to know what is generally speaking happen. You start as a membrane and you start to calcify, and examples. The blood vessels here are also important. The nutrient, where is it located? And the metaphyseal arteries. Effect of exercise and lack of exercise. Just the basics. Exercise more, the bone is stronger, less, the bone degenerate. What also is important besides calcium and phosphate? You need some magnesium, fluoride, iron, and manganese. It's very important to remember here the rule of parathyroid hormone, calcitonin, and calcitriol, which is different. Okay, parathyroid hormone, vitamin D, which is cholecalciferol, and calcitonin and calcitriol, and do not confuse those because all of them, most of them start with cal. So calcitriol, calcitonin, calciferol. The three of them start with cal, so you can very easily confuse those. Do not confuse them. They are completely different. Calcitriol, that will make the vitamin D3. Calcitonin is an enzyme that decreases calcium. Parathyroid hormone increase calcium. Okay, this slide is very important too. You have to know what exactly is needed for the bone growth and what does it do. Like C for collagen, stimulating osteoblast. A for osteoblast. K and B12, proteins. Growth hormone, stimulate the bone growth, and thyroxine too. Estrogen, stimulate osteoblast. Calcitonin parathyroid, regulation of calcium and phosphate. So this is important. What is the difference between dwarfism, giantism, and acromegaly? And I actually asked you about that, and a lot of you did it wrong, as far as I remember. So you need to remember that. Well, only when I give you the clue, if you have a lot of growth hormone. But what's the difference between giantism and acromegaly? What's the difference between those two? Before and acromegaly? After. And both of them are overgrowth, right? So you have to remember, it's all about after or before the bone growth. What's the function of calcium? We mentioned the bone. It's also neurons, muscles, and muscle contraction and membranes. Again, parathyroid hormone, calcitonin, calcitriol. What is the difference between those three? It not only whether they increase or decrease calcium, how? And remember the drawing that I did here? Intestine versus the bone versus the kidney. So this is very important to remember. And where is it secreted from? Like calcitonin is secreted from C cells or parafollicular cells. The parathyroid hormone from the parathyroid gland. Calcitonin, thyroid gland. Which cells? C cells, specifically. You need to remember that the normal level of calcium, 8.5 to 11. For fractures, remember the steps. Start with bleeding, endosteum and periosteum start to repair. What is external and internal callus means. And then the rule is of osteoblasts and osteocytes in repairing. For each one of these fractures, you need to know the basics. Pot fracture, which is both bones at the leg. This is what you have to know. Comminuted fracture, when you have shattered bone, basically. Transverse fracture, you break something transversely, like the ulna. Okay? Spiral fracture, like twisting, something like the tibia. Displacement fracture is when the bone is misaligned, misarranged. Coolest fracture is when you fall out, to cushion to fall, and this is usually the distal part of the radius. So just the basic. Green stick, children. And it's usually one side is broken and the other one is bending. That's all what you need to know. Okay, epiphyseal fracture at the epiphysis. What's the problem? Stop growth. That's what you have to remember. Compression fracture is like if you fall down seated in seat position, and this is in the vertebrae. Osteopenia. What are the types of osteopenia? We talked about rickets versus osteomalacia and osteoporosis. Osteopenia is just weakness in general. Osteoporosis is something else. This is severe, more in females, and it's due to hormonal problems. And the bone become porous, fragile. Okay? Aging, everything deteriorates. Okay, chapter 9 is about articulations. This table will summarize everything for you. You have to memorize this table, repeat it as many times as you want. But you have to memorize this from all aspects. I can either ask you what's gomphosis, what's synarthrosis, and what's the degree of function, and what are the examples. You have to memorize this table. You don't get mistakes. It's 2 parts. You have to memorize it from all aspects, not just one way. Meaning, don't expect that I have to ask you, well, what's arthrosis? And you answer this. No, it can go the other way around. I can ask you, the joint between the tibia and fibula is amphiarthrosis, diarthrosis, or synoarthrosis. For example, is it syndesmosis, symphosis, monoaxial, biaxial? So you have to study it from different ways and ask yourself the questions. You can start with the example, you can start with the description, you can start with the function category. So you need to memorize those two pages, those two tables very much, very important. Okay? This is just a repeat, so the first table is very, very important. Diarthrosis is like synovial, and we need to know the synovial in details. Okay? So if you talk about the synovial, which is diarthrosis, but the structure itself is important. So it's surrounded by synovial membrane, synovial fluid. What's the function of the synovial fluid? Lubrication, reduce friction, nutrition, okay? And the accessory structures, what's the function of each one of those? What's the menisci? And expect something like sprain. Sprain, for example, versus strain versus dislocation versus subluxation. These four conditions are actually important. You have to understand the meaning of each one of those. Sprain, strain, dislocation, subluxation. Dislocation is also called luxation. What's the difference between tendon and aponeurosis? What's the bursae? Okay. This picture is actually important because it summarizes what you need to know. Okay, what all these factors will stabilize. So this picture is very important. I'm not going to give you a picture, but it's important to remind you with the different parts and different structures of the knee specifically, because this is the only one that I want you to know the details of the structure, which is the knee. So dislocation, subluxation. I mentioned before that from 22 to 45 is not very important, so let's skip this. You can read it if you need to. Flexion, extension, abduction, adduction, this is all lab. Okay, these tables are important. So all these, what we skipped already, this is all lab. Don't expect much question from these, but this table is actually important. Here is one, the type of movement and the example. 1, 2, 3, and then you have this table, another 3. So you need to know the type of joint, is it biaxial, monoaxial, or triaxial, and the example. Okay? Intervertebral disc is very important. You have to understand this. What's the function? And what is the difference between what's annulus fibrosis versus nucleus pulposus? And what if any one of these is herniated? It's not the same thing, as we know. Okay, the only ligaments that are important just to know the name are the intervertebral ligaments and the ligaments of the knee. Any other ligaments, you can read it, but I'm not going to ask you about it. Okay? Here it is, slip disc versus herniated disc. This is important to know the difference between those two. It's not the same. And it's not the same consequences. So this is annulus fibrosis, slip disc, herniated is nucleus pulposus. This is one. Number 2, effect, vertebral canal or spinal cord and nerves, herniated disc. Okay? The names. Yes. So you need to know the ligaments for the vertebral column and for the knee. Otherwise, you can read it, maybe for the lab a little bit, but not for the lecture. The types of joints, just the main thing. Is it diarthrosis? We mentioned that before, like ball and socket. This is the shoulder and what's the other joint that's ball and socket? Hip. And why this is the most frequently dislocated? Shallow. What? Because of shallow. What? Glenoid cavity. Knee joint, you need to know the details about the knee joint. Okay? It's two joints in one, two in one. Articulation, two articulation, medial and lateral condyles. You need to know the details of this, including the menisci, what's locking of the knee. So the details about the knee joint is important. Everything about the knee joint. Okay? Including the ligaments. All other ligaments, as I mentioned, is something for you to read, but details needed for the knee. These conditions are important, of course. What's rheumatism, arthritis, osteoarthritis, rheumatoid arthritis, gout arthritis? What happened in joint immobilization and aging? So these 1, 2, 3 slides, very important. Okay? And this also is important, the bone recycling versus bone strength. What are these factors here? So this slide is also important. Okay, so that's it for this one. Okay, this one is important here. You have to differentiate between epimysium, perimethium, and endomysium. And just what's the difference between each one of those? What does it surround? Okay? Is it surrounding the individual muscle cell, bundle, which is fascicle, or the whole muscle? Okay? What's the difference between tendon and aponeurosis? It's important. And how we make the tendon, fusion of the three. Okay? We just covered that. We talked about this a little bit today. How do we make the skeletal muscles? And why is it large, huge diameter, and a lot of nuclei? Because it comes from fusion of myoblasts. What's sarcolemma and sarcoplasm? What's the function of the transverse tubules? Internal organization, what's the myofibers? That's what I talked about yesterday, starting with the whole muscle, going to the fascicle, to the muscle fiber, to the fibrils, to the filaments, which one contain actin, which one contain myosin. We covered all of that. What's the terminal cisternae, what's the triad? And the process starting from releasing of, the arrival of the action potential to the nerve terminal, and the process that occur from this point until contraction and relaxation. That's all important, and I think I covered that in details. What's the M line, Z line? What's the H band? What's the function of titin? What's the function of the sarcomere? Where is the calcium produced or released? Again, I mentioned that. starting from the arrival of the action potential to the nerve terminal, to the calcium entering, to the acetylcholine in the vesicles open, the vesicles move, the acetylcholine is released, crossing the gap, interacting with the receptors, opening for sodium, action potential in the motor end planes, and going to the sarcoplasmic reticulum, releasing calcium, attaching to the troponin, moving tropomyosin, uncovering active sites, coupling of the active site with the heads of the myosin, starting the contraction, release, recover. Okay? Very simple. You have to study it, and I explained it so many times, so you have to study it. I never said this is easy, but you have to. Okay, so what's the function of each one of those? F-actin, nebulin, tropomyosin, and troponin, and the whole process. Okay, so we discussed that. The details are actually needed. What is excitation contraction coupling? I mentioned that several times. Important. What happened at the synaptic terminal, including the function of acetylcholine? What's the enzyme that breaks it down? What if you don't have it? Okay? What's the name of the enzyme? What's the name of the enzyme that breaks down acetylcholine?
ACHE.
Yes. Excitation contraction coupling. This is the 5 steps to contraction. It's important. Exposure active sites, cross-bridging, pivoting, detach, reactivation. Factors determine the duration of stimulus, number of calcium, and ATP. How does the relaxation occur? Important. And what's rigor mortis? Important. What's the all-in-one principle? Okay. What determines the tension of one muscle fiber? The number of cross bridges pivoting, the resting length, and the frequency of stimulation is also important. All-or-none rule is important. We mentioned the length-tension relationship, which is specifically important in the skeletal muscles. What's twitch? Okay. Here's the latent period. Do you remember the latent period? The latent period, contraction, relaxation phase. And obviously, TREPI, wave summation, incomplete tetanus, complete tetanus. The 4 stages are important. TREPI, wave summation. And the time is also important. More than 50 seconds in wave summation, less than 50 per second in the rest. I mean, less than 50 in TREPI and more than 50 per second in the wave summation, and then incomplete and complete tetanus. What's the difference between each one of those? What's recruitment, and how can we achieve the maximum tension? What's the muscle tone? What's the difference between isotonic contraction and isometric, including concentric versus eccentric? Isotonic versus isometric. And isotonic, specifically concentric versus eccentric. Is the tension more or low, or the load is more? That will determine those two. Versus isometric. Okay, what's creatinine phosphate? Okay, and can it be used, or we have to change it to ATP first, right? What's the difference between aerobic and aerobic? And actually, I explained that more in the next part, so you can take it from there. How many, including how many ATPs? How many ATPs do we get from aerobic?
34.
And in anaerobic?
2.
2. Okay, here it is. This is the process that occurred, and that was here. That was important. I draw it. If you got that already, that's what you have to know. From the process, glucose enter, aerobic acid, glycolysis, to, okay, this is important. Or you can take a look at this picture, it will explain it too. Here is the use of fatty acid versus lactic acid. But if you draw what I'll draw here, that should give you what you need to know. Including the production of lactic acid, lactic acidosis, Curie cycle, right? PCA. This, what I put here, is the most important thing to remember. What's the muscle fatigue? What's the need of recovery period? And what's the Curie cycle? Oxygen debt. Curie cycle, lactic acid change to pyruvic acid in the liver. What's the oxygen debt? Heavy breathing after you stop exercise to cover for the debt. Oxygen debt. What are the hormones that affect the muscles? Important. Growth hormone, testosterone, thyroid, and epinephrine, and I will discuss the details of this. How to modify the power and endurance, and the difference between these fibers, which is this table. So this table is important, table 10.3. This is very important, including the other names. Type 1, which is red, slow. Type 2A, which is pink. Type 2B, which is white and fast. So this table is very important to memorize. That will cover you from all aspects that you need to know. What's the difference between hypertrophy and atrophy? And we're talking about the physiological point of view. So we're talking about like muscle building versus you're laying down, not doing anything. Hypertrophy versus atrophy. Examples of anaerobic, like the 50-meter dash. Example of aerobic, like light exercise. Cardiac muscles, the difference. And there is a table actually that, I think there was a table that summarized those three together, and this should be enough. Which is small, which is large, which is striated, which is not striated, which is one nucleus, which is multinucleated, and why. Do you have a triad or not? Do you have terminal cysteine or not? Including intercalated discs. Intercalated disc in the cardiocytes. This is also important for automaticity. So this slide is also important. Automaticity, which is in the cardiac muscle, and pacemaker, which is the pace setter in the smooth muscles. Smooth muscles are not striated. These characteristics are important, but you can actually just jump to the table that will give you what you need to know. Excitation, contraction, coupling here is different, and I told you, and this is important. Where it's the location of calcium, most of it is in the cytoplasm, a little bit in the sarcoplasmic reticulum, and instead of binding to troponin and the skeletal muscles and cardiac muscles, it actually reacts with calmodulin. And this enzyme, you need to remember, myosin light chain kinase. Okay, so this table is very important. 4B. This is summarizing the differences between skeletal muscle, cardiac muscle, and smooth muscles. So that should cover you from, this is all what you need to know about the comparison between the three. For chapter 11, there's just a little part, just a couple of slides that you need to know, which is the organization of the muscle fascicles. The organization can be parallel convergent, pinnate, or circular. And the most important thing here is the example, like parallel. The example for that is biceps brachii. The fibers are parallel to the long axis of the muscle. So this is the biceps, which is an example of the parallel arrangement of the muscle fibers and muscle fascicles. The convergent is like this. This is the pectoralis muscle. This is convergent. The fibers are converging to the tendon. And the example again is pectoralis muscle. The next is pinnate, and pinnate can be uni or bi or multiple pinnate. Examples: unipinnate, extensor digitorum muscle. Bipinnate, the example is rectus femoris muscle. The multipinnate, the example is deltoid muscle. And the last arrangement is the circular arrangement of the muscles, like the sphincters, for example, like the orbicularis oculi, orbicularis oris, or any sphincters in general will be arranged in circular arrangement. So again, the muscle or the fascicle arrangement and examples of that.